So our next speaker is Kory Lavine. He's a professor at WashU in St. Louis, and he's going to be speaking to us about his bispecific T-cell engagers for various cardiovascular, fibrotic diseases, etc.
So I'm going to talk today about some of our work on cardiac fibrosis, and what we're really going to touch on is roles for bispecific T-cell engagers in diseases outside of oncology, outside of inflammation, and some of the things that we've learned about trying to apply these really amazing molecules in areas that they really weren't invented to work in. And so we've learned a lot of interesting lessons that have been very informative of how to move forward, and so I'm really excited to share that with you. All right, so why go after cardiac fibrosis? So I'm a heart failure cardiologist. We see these patients all the time. And as many people would expect or understand, individuals with coronary artery disease that develop heart failure have a lot of fibrotic material in the heart. And that's shown in these typical H&E images as this kind of pink frothy stuff between adjacent cardiomyocytes, which are in the darker pink or purple color. But it's actually very prominent across all forms of heart failure, including non-ischemic cardiomyopathies, genetic cardiomyopathies, cardiomyopathies due to hypertension, and represents a really critical area in disease that we really have no therapeutics for. And so we know that fibrosis is not only present and correlates with outcomes, but it's actually causative. And so there's been multiple Mendelian randomization studies, as well as these really interesting prospective cohorts that have used cardiac MRI to gauge how much fibrosis there is in the failing heart, and have shown across thousands of individuals and many studies, in addition to this, that individuals that die of heart failure have much more fibrosis than those that do not. And in fact, if you see a patient with heart failure and no fibrotic material in the heart, they're much more likely to recover with standard therapies. So today, there's a lot of work in the fibrotic landscape across kidney disease and lung disease, but as you can tell from this graph, there's really nothing in the heart. The only square here is pirfenidone, which we know is really not effective. So there's an open therapeutic opportunity to target cardiac fibrosis that really is not being pursued. And one of the key reasons that it's not pursued is we don't really understand what the type of fibroblasts that drives fibrosis might be and how to target it. And so we decided to take this on a few years ago. We have access to lots of human tissues, which is a fantastic thing. And we're able to profile hearts that were not diseased, that were not used for transplant, and compare that to individuals who had to have a heart transplant because they had a large infarction, or they had chronic forms of non-ischemic and ischemic cardiomyopathy, and we're able to deploy multiple forms of single-cell sequencing, including site-seq, which allows us to see the surface epitome of cells by using conjugated antibodies, as well as multi-omic sequencing that allows us to understand more of the transcriptional landscape and how gene regulatory networks are put together by assessing chromatin accessibility. So in doing that, we were able to make nice maps of all the cell types that exist in the failing human heart. This is an example of one of those. It's a UMAP plot. Each dot's a cell. The cells are clustered. The closer they are together, the more likely they are to be an individual cell type. And you can identify the cell types based on canonical genes that are differentially regulated in each of those clusters. And so using this, we could identify fibroblasts and endothelial cells and myocytes and pericytes and all the important cells in the heart, and then really minor populations like B cells, plasma cells, and mast cells. Today, I'm going to focus on fibroblasts, which is shown in the red color. And as you can see in the bottom pie graph, that compared to donor hearts, patients who had an acute MI, that's AMI, ischemic cardiomyopathy or non-ischemic cardiomyopathy, have an expansion in the red population of fibroblasts. And if you look at amongst all the differentially expressed genes that are present between a donor heart and a failing heart, the majority of differentially expressed genes are actually within this fibroblast population, which tells us that there's really a lot of events going on there and a lot of changes. So what's going on in the fibroblasts? So we can individually cluster these cells, as you would if you're clustering a cancer cell or cells within the tumor. And I'm not going to go through every cluster and bore you, but I'm going to talk about two of them up front, and then one really will be the focus of the talk. And so if you look across the different disease states between control patients or donor and the different forms of heart failure, there's really two fibroblast clusters that really expand and emerge. So the first one is in dark blue, which are myofibroblasts, which are the smooth muscle actin expressing fibroblasts that a lot of us think about when we hear about active fibrosis. There are pretty few of them in the donor heart, and they expand across each of the 3 diseases. But unexpectedly, we identified an additional fibroblast population that's really not at all in the donor heart, and that's in the brown and it's circled up there. It expands dramatically in acute myocardial infarction, remains elevated in chronic forms of heart failure. And this population is marked by fibroblast-activated protein, which is a fibroblast protein that's known to be in cancer-associated fibroblasts. It co-expresses periostin at the RNA protein level, and then also expresses markers of activated fibroblasts like Ty1. So it's a little bit of an unrecognized active fibroblast population. And if you look back at bulk RNA sequencing data sets of other forms of heart failure, such as hypertrophic cardiomyopathy and genetic forms of cardiomyopathy, FAP was actually one of the top differentially regulated genes. And so it's really there across all forms of heart failure. Okay. So the important point I want to make in this slide is that these FAP-positive fibroblasts are not myofibroblasts. They're in a different location and have a very different function. We can do inferences from trajectory analysis that show us that the 2 inferred end states of activated fibroblasts are myofibroblasts, which is shown as F2, or smooth muscle actin expressing. And then there's a separate one, which was F9, which is the FAP and periostin expressing cells. If you do immunostaining, they're completely non-overlapping. The FAP and periostin cells are in white, and the smooth muscle actin cells are green, and there's absolutely no overlap. And then we can take additional data sets and show that they're located in different positions within the myocardium. The myofibroblasts are located within the core of the infarction and play roles in structural integrity. And then the FAP and periostin expressing fibroblasts live in the periphery of the infarct on the outside, where there's a lot of inflammation and active remodeling. And we can make mice where we can tag the FAP expressing fibroblasts and follow their trajectory. We can do the same thing with the myofibroblasts, and they don't overlap at all. So these aren't transient states that intermix. They represent distinct populations. And on the bottom is an example of our FAP lineage tracing. The FAP cells are shown in red, periostins in white, and smooth muscle actins in green, and there's absolutely no overlap between those populations, which is quantified on the right side of the screen. So what's the information that these cells are pathologic? And so one thing we can do in mice is we can remove them at the time of cardiac injury, in this case, a reperfused infarction, which is similar to what a patient experienced in the cath lab. They would come in with a heart attack, where they would get a stent put in, which reperfuses the vessel, and we simulate that in animals, and then we depleted this FAP-positive population. So with the genetic depletion of these cells, we can achieve about a 75% reduction. The cells are in red. You can see at the top is a control and the bottom is a depletion, and we can see a nice reduction. And if we look at these animals 4 weeks after the infarction, and you don't need to be a cardiologist to see this, the hearts on this side have a nice infarct at the apex. The heart dilates and the function reduces. And when we remove the FAP-positive fibroblasts, the infarct's a little bit smaller, but the volumes and function really are a lot better. And when you look at the quantified data, there's absolutely no overlap between groups. The ejection fraction, meaning how much blood is removed or is ejected with each beat, is higher. The size of the chamber is reduced, and the size of the infarct relative to the initial infarct size is reduced as well. So we have a positive outcome. This tells us that this is a population of cells that we want to remove and that could have therapeutic benefit. None of the animals died, none of the hearts exploded, so it was not, important for structural integrity. If you removed myofibroblasts, you would see rupture and you would see death in the mice, and so that's an important point, even though I'm joking about it. All right, so we've looked in the laboratory and moving forward therapeutically, but how can we target these cells? And so we've learned a lot of things. We learned that they're specified through an IL-1 pathway that goes from CCR2 positive monocytes and intermediate macrophages, not fully differentiated macrophages, that signals directly to the fibroblasts. That's important for their specification. We've identified transcriptional pathways regulated by RUNX1 that mediate their differentiation. But I'm going to talk today about a different therapeutic approach we thought about, was trying to remove them with a T-cell engager. And so we did this work in collaboration with Amgen. So these are their reagents. And so we used an anti-mouse FAP antibody that is about 15 to 20 picomolar KD. We use an anti-CD3 arm, which in the bispecific format has about a 25 nanomolar binding constant. So we have nice affinity where we have much higher affinity to FAP than we do to CD3. When we make a target cell line that expresses FAP, and we can robustly remove the FAP positive cells, and the EC50 of this is all the way down to 50 to 75 picomolar. So it's a really potent reagent that it works really well. And so we deploy this into mice, and so when we do that and look at our FAP positive cells, we see a dramatic reduction, something like 90, 95%. But when we look at the animals after their infarction, we saw something totally different. So this is a control, and then this is an animal that received the FAP bite. And you can see that the heart really got really big, the function got really poor, the infarct got larger, and a few animals died. And so we had the opposite effect than we would expected based on the genetic data. And this is what this is the quantitative data, very tight in terms of reduction in ejection fraction, and increase in akinetic area relative to the initial infarct size. And we look at the pathology that bears that as well. And red is the fibrosis. And you can see that there's a lot more fibrosis and dilation of the FAP bite exposed hearts. So what in the world's going on? So one way to conceptualize this is we think about these T cells as their main role is to kill, right? And they likely do this through perforin and granzymes and death receptors. I'll go through that in a second. But when these T cells get activated, they do a lot of other things, right? They make interferon gamma, which I'll show in a few minutes, and they make other mediators that can communicate with the rest of the environment. Things that we don't really think about that they probably do in the tumor microenvironment, but we're really seeing that within the myocardium. All right, so we decided to characterize this to understand what is actually happening. What are these T cells doing? So we initially did bulk RNA sequencing, and when we did the bulk RNA sequencing, we found one pathway that stuck out like a sore thumb, and that was interferon gamma signaling was up literally like a zillion-fold. It was night and day between groups. Interferon gamma itself was up 40-fold, CXCL9 was up 40-fold, and other markers were 5-fold and 10-fold, et cetera. So lots of interferon gamma activated genes. If we look at where T cells might be, we saw that in the control, compared to the control, the FAP bite treated group, that there was really a large accumulation of CD4 and CD8 cells that were present within the border zone where these FAP cells presumably would be. And that was markedly increased when we quantified that. And those cells express a lot of granzyme. They're a mix of CD4 and CD8, as well as a number of interferon gamma-associated transcripts and other transcripts associated with T cell activation. So we then performed single-cell sequencing to understand what these cells might be and how the bite changed their transcriptional state and their behavior. And so I'm going to focus on two populations today. I'm going to focus on the T cells, which I think that's probably one of the most interesting things. And we learned that those T cells will communicate with macrophages, which I won't talk much about, but also will communicate directly with fibroblasts. Okay, so what happens with the T cells? So in a typical control heart after myocardial infarction, we see a lot of different T and NK cell populations. We see NK cells, we see memory T cells, and a small population of CD8 T cells. When we get the FAP bite, we saw a dramatic shift in the relative proportions of those populations. We saw an expansion of CD4 T effector memory cells, and we saw exhausted and toxic CD8 T cells really expand. Thank you. That's nice of you. I appreciate it. And when we look at where the interferon gamma is coming from, which is in the purple here on this dot plot over here, the majority of interferon gamma actually is coming from CD4 cells and those activated CD8 cells. And these T cells took on a Th1 type phenotype, not a Th17 or Th2. It looked like a Th1 phenotype based on TBX21 interferon gamma. And then they also express a lot of CD40 ligand, which would be predicted to activate macrophages and potentially maintain an autocrine loop of interferon gamma signaling. Within the macrophages, we also saw interesting changes, and we saw a specification of a interferon gamma population of macrophages, which is shown here. And then we also lastly saw some really cool differences in T cells. So within the T cell compartment, the reason we saw more fibrosis is we had a switch in the activated T cells. So this is spatial transcriptomic data. I put it here instead of the single cell data for sake of time, but also because it really exemplifies the point. In red are the FAP and periostin cells that are present in the control heart. After infarction, you can see they all localize to the infarct zone. And when we give the FAP bite, they're pretty much all gone. We see about a 90% reduction at the spatial sequencing or the single cell level. But instead, we see a new population of fibroblasts enter this area. These cells are marked by interferon gamma responsive genes. They express periostin, but they don't express FAP. And so we found another pathway to get an activated fibroblast that we perceived may be interferon gamma regulated. So to test that, and understand the role of interferon gamma and potentially CD40 ligand signaling in these FAP bite associated pathology, we started with our in vitro system, and we asked the simple question of, does interferon gamma and CD40 ligand is important for killing within this assay? So here we take T cells, we take our bite, we take our target cells, and then we assayed over different concentrations of bite, and we can see a marked increase in interferon gamma on the top row compared to the control. And then we see increased percentage of CD40 ligand positive T cells as well. So both of these things can be recapitulated in vitro, and they have absolutely no bearing on killing. So when we neutralize interferon gamma with a huge surplus of neutralizing antibody or CD40 ligand or the combination, killing remains at 80 to 90%, which is about the number of cells that were expressing FAP. Instead, killing was dependent on trail, which is shown in the latter graph, as well as dependent on perforin and granzyme. So we think the killing is a different mechanism than interferon gamma and CD40 ligand. And so to prove that in vivo, we took mice that were infarcted, we gave them the FAP bite, and we either gave them isotype control antibody or neutralized interferon gamma and CD40 ligand signaling together or separately. I'm just going to show the together data. When we neutralize them together, we can essentially neutralize the interferon gamma response and reduce the amount of ligand that's produced, which probably represents this feedback loop. This has no effect on killing. We still can kill all of the FAP positive cells despite neutralizing these pathways. And now we can rescue the phenotype. So here's the control bite. The control bite when you neutralize interferon gamma and CD40 ligand. And so these pathways are not normally activated after an infarction, so there's no difference between these hearts. If we give the FAP bite with an isotype control, again, we see a worsening in the cardiac phenotype. And if we neutralize interferon gamma and CD40 ligand, not only is it better than the FAP bite alone, we now see a therapeutic response where it's better than the control, similar to the genetic depletion. And this is the quantitative data. If you focus really just on ejection fraction for simplicity, no difference in the control bite, whether we give isotype or the neutralizing antibodies. With the FAP bite, the function goes down, and then we neutralize interferon gamma and CD40 ligand pathways, the EF goes up, and it's now greater than the control. And so we were able to unmask the therapeutic effect by blocking this sort of collateral pathway. And just to finish and prove that this is related to interferon gamma signaling to fibroblasts, we deleted interferon gamma receptor in fibroblasts in the presence of the FAP bite. And so when we do that, we can again see an improvement in cardiac function with the FAP bite and a reduction in infarct area. And so we're able to, again, unmask the benefit by removing a signal of interferon gamma to the fibroblasts. So in conclusion, I hope I convinced you that depletion of FAP-positive fibroblasts could represent an important target. It decreases infarct size, improves cardiac function after MI. FAP-targeted T-cell engaging molecules successfully deplete target cells in vivo after cardiac injury, but they surprisingly worsen fibrosis and dysfunction. The FAP bites induce an interferon gamma expression in T cells, which then signal to macrophages and fibroblasts. And the neutralization of this pathway was sufficient to improve, reduce inflammation, reduce fibrosis, and unmask the therapeutic response. And so we think this gives us really important clues about how we should target T cells in the heart, what T cells we might want to engage, and what pathways we don't want to stimulate if this type of therapy would be used in the cardiovascular setting. So a lot of people to thank. My laboratory on the side in red are the people that contributed to this project. And then I'd really like to take a second and thank the Amgen group and CMD, which really made these studies possible with the reagents and device. So thank you so much, and I'd be happy to take any questions.
Very cool study. Just wondering, have you looked into the bone marrow toxicity with your Fab T-cell engager?
Yeah, great question. It reminds me of a point I didn't bring up. So when we gave the FAP bite in this setting, not only is the inflammatory response, it's restricted to the heart, so we don't even see interferon gamma in the blood, and then the bone marrow is not affected at all.
You don't see any,
No, so all the major blood counts are the same, and all the hematopoietic progenitors are there at the same abundance and amount. And I think a key reason that we're seeing that sort of phenomenon is that the FAP arm is so tightly bound to its target that we think we're engaging T cells only within the heart and not in other areas of the body.
But bone marrow has a pretty high FAP expression.
Yeah, well, we're not seeing that. So we do have some biodistribution studies trying to look at where the FAP bite is going. And we actually did not detect that much in bone marrow. The majority, for whatever reason, was going to the heart. Maybe that relates to accessibility, I'm not sure, or just the abundance of antigen in the heart.
Okay. I think you probably know Amgen used to have a bite to target Fab. They stopped.
Say it again?
Amgen used to have a bite to target FAP for cancer.
Okay.
I think they have a very severe tox, I remember correctly.
Yeah, well, the data is the data.
Okay,
so,
thank you.
Yeah.
Thank you for the talk. That was really interesting. I was wondering, have you considered making a tri-specific to block the interferon gamma receptor, given like the data on your last slide?
Yeah, exactly. So we're looking at different approaches where instead of having like an add-on on the target, to do that on the T cell. So one thing we learned from these studies and have time to talk about is we put a DNA barcode on the bite to ask what T cells it's actually engaging. And it's clearly only engaging T cells within the heart and not in other areas. Mm-hmm. And so that tells us the T cells that are present really make a big difference in the response. So we're looking at approaches that we can modify the T cells that are there, or selectively only engage other T cells like CD8 T cells that kill but don't have this as a robust interferon gamma response as the CD4 T cells do.
Cool.
Great question.
You.
Hi, very nice talk, thank you. Just a question about the gamma mechanism. Do you think this could be possibly an attribute of the particular CD3? So is it the killing itself, or is it the strength of signal of the CD3? So say with a lower affinity CD3, might you be able to induce that killing in the absence of cytokine production?
We don't know till we do that experiment, right? We did try to pick something that was much more tightly bound on the FAP arm than the CD3 arm. We have dosed at different amounts to try to indirectly affect that. So the experiments I showed you are dosed at 10-fold above the EC90 at trough. We've also dosed it such that it's 1x EC90 at trough, and we see the exact same effect. So that's kind of addresses your question, but I think the only way to do it is really have a lower affinity antibody. We're doing those experiments now. Thanks.
Okay, thank you very much. One final question. What about the fibrosis that's in atrial fibrillation?
Yeah, it's great. That's a great question.
You think that's...
Same fibroblast population. So this is a fibroblast population that is derived from resident fibroblasts when they get activated, and it has a similar signature no matter what chamber of the heart you're looking at.
Got it.
Thanks.
Okay, thank you very much.